Hello. How are you? Grade 11 biology, unit 3, about enzymes. The unit is a very splendid unit, please. Today, I'm coming with one aspect of this enzymology, please. Before I start my lecture on enzymes, please subscribe, like, and share my videos, please. Also, your comments, put your comments so that it may help us for growth together. Now, let me go straight into the lesson, please. Today's topic video is on properties of enzymes, a very important aspect of enzymology, the science of enzymes. Uh, Properties of enzymes, um, general properties of an enzyme. Of course, we may see the properties of enzymes by classifying into chemical and physical properties. Of course, this is the general properties of an enzyme. The general properties of an enzymes are the nature of both physical and chemical properties combined. When we combine the physical as well as chemical properties, we call it general properties of an enzyme. Therefore, here enzymes, one aspect, one property is generally speaking, enzymes accelerate or speed up the reaction rates. This is one property. What is the biological role of enzyme? Of course, this is the role they assign to these special groups of protein molecules. Of course, enzymes accelerate or speed up or increase reaction rates. Therefore, they serve as catalysts. They neither affect the nature of products formed nor undergo any changes by the reaction catalyzed. Therefore, they are serving as catalysts. They don't affect the nature of product formed or they don't undergo reaction in the process of catalysis. That means this is a special property of catalysts, of course. The other general property is an enzyme possesses specialized region. You see, it's a protein molecule has a specialized region, which is known as active site, or it is a cleft or gorge. Even if you like, some biologists dub it or give it a nickname, the vagina of the enzyme, the cleft, the gorge, special pocket that, to which specific substrates bind or attach. That is chemical binding, of course, through different types of bonding. Therefore, an enzyme possesses a specialized region. It could be one or many regions of core active sites, uh, special pockets in the spherical enzyme where the specific substrate attach and the catalysis occurs. Uh, Under high heat, temperature, and varying pH, an enzyme becomes unstable, or we can say that some environmental factors affect the activity of enzymes, and these environmental factors are heat, temperature, and change in pH. Of course, then you have specific heat, they can function properly or efficiently, a specific temperature at which they can function properly, and a specific pH range in which they actively function with no problem efficiently. Therefore, there are some factors which affect the rates and the activity of enzymes. The other property, enzymes are proteinaceous, possessing properties, characteristics to proteins. They are proteinaceous. Of course, they are proteins, except, of course, some RNA molecules which serve as enzymes. A small amount of enzyme is required to bring catalysis of substrate. You see, enzymes are protein molecules, exception ribosomes. I will come to this point later. Otherwise, they are proteins made from amino acids by gene expression process. A small amount of enzyme is required to bring catalysis of substrate. Usually, we require enzymes in small amount because they are highly efficient, so that they are needed in a very small amount inside the body. Enzymes can be used and reused again and again. Why? Because they are not changed at the end of the reaction. We say this is also another property. And let's keep on mentioning the properties. Enzymes activity typically shows absolute, relative, group, or stereo specificity. You are there specificities. Generally speaking, we can say enzymes are highly specific, and this specificity can be divided into absolute specificity, relative specificity, group specificity, and stereo specificity. Later on, when I come to discussing specificity, I will discuss about the different types of specificities of enzymes. Of course, some enzymes are regulatory in function. Of course, yes, some enzymes regulate, for example, regulate gene expression, that they bring about change in the genes or sequence of nucleotides in a DNA, so that either help in keeping the gene shut off or help in the gene to be expressed. They can be used to regulate gene expression. Some enzymes are even used to regulate other enzymes. The activity of other enzymes can be regulated by the action of some other enzymes. You see, some enzymes are regulatory in function. They regulate biochemical processes in our body, so that homeostasis is possible, so that a product is produced as needed by the body, so that a gene is not expressed if its product is not needed, or the gene is expressed if its product is needed by the cell. Its primary function is to minimize the activation energy. Enzymes primary function is to minimize activation energy. If you are asked, how do enzymes bring about acceleration or increase in speed of reaction or rate of reaction? Just by decreasing the activation energy the substrate needs in order to convert into product. They bring about decreased activation energy required by the substrate. When substrates require a small amount of activation energy, this small amount of activation energy is available from the environment in the form of it easily. It is available for most of the substrates, so that many substrates can change its product within unit period of time, so that many products are produced by due time. If the activation energy that substrates require is huge or large, only very few substrates can get this huge activation energy requirement, so that only few substrates change the product, which means very slow rate of reaction. Therefore, if you are asked how do enzymes speed up reactions, under this, they speed up rates of reaction by decreasing the activation energy that substrates require, so that many substrates can get that small amount of activation energy from the environment and convert into product. That is very nice of these smart molecules, really. Uh, the other property, the enzymes. Enzymes remain unaltered during and after the product formation. That means I have already mentioned this property. Of course, enzymes are unchanged at the end of the reaction, or they don't bring change in the product. They don't change it themselves. So, if they are not changed at the end of the reaction, they catalyze, so they can be used again and again. We can use them, of course. Are we using them forever? No, no. Enzymes can also, because of service for a long period of time, they can become with no normal. They may lose their three-dimensional specific shape because of the wear and tear, aging. You age. So because your proteins age, when we say proteins age, yes, enzymes also age, because the wear and tear, because of long-term service, they may lose their three-dimensional specific shape, including the active site, so that they will not be effective in catalyzing reaction, so that they are replaced. They are recycled. Their amino acids are recycled. They are broken down. Their amino acids are recycled, and the gene that is producing these enzymes will be on and expressed and produce the enzyme, that specific enzyme, in order to replace those worn out or aged enzymes. Otherwise, until they age, they can be used again and again since they are not changed at the end of the reaction. They catalyze, of course, yeah. An enzyme may possess an allosteric site besides an active site to which cofactors or regulators interact. You see, we have talked about specific pockets or the specific left, if you like, that specific vagina of the enzyme where the substrate binds. In addition to this special site, the active site, some enzymes do have allosteric site. Allosteric means other site. Allosteric means other side, other than the active site. This allosteric site is a place where allosteric regulators or allosteric modulators, which are substances which attach to the allosteric site, so as to regulate the activity of enzymes. Therefore, these allosteric regulators or modulators could be one allosteric inhibitors, which attach to allosteric inhibitor site and stop the activity of enzymes, and it could be allosteric activators, which attach on the allosteric activator site, so that they inhibit, they stimulate the enzyme, and the enzyme becomes active, and the product is produced in large amount. Therefore, the allosteric site is could be a place for attachment of cofactors or regulators. We shall talk what cofactors are. Cofactors are non-protein molecules or metal ions which are uh, which attach with enzymes so that the enzyme can be active. There are some enzymes which require non-protein part. This non-protein part, which is required for active enzyme, generally are known as cofactors. They could be, of course, organic like coenzymes, prosthetic groups, organic these also, and also metal ions, some metal ions like magnesium, zinc, manganese, all these metal ions. We shall talk about these cofactors in detail in the next upcoming slides, of course. Now, let's go. Enzymes are soluble in water and sodium chloride, salt solution, of course. Yes, they are globular proteins. When we say enzymes are proteins, and the type of protein they represent is globular protein, which are soluble in water and also sodium chloride solution. And now let us see in detail the physical properties of enzymes. Physical properties. Now, the physical properties of enzymes include denaturation, losing its three-dimensional shape because of changing pH, because of changing temperature, because of some other denaturing agents, of course, chemical denaturing agents. The other physical property, solubility, able to solubilize in water. The other is colloids. They are colloidal in solutions. They are colloidal. That means colloid means they can easily pass their huge molecular weight. Enzymes do have very big molecular weight, so that they can't come out of membrane easily. That gives them the property of colloidal nature. They are biocatalysts. Precipitation, they can precipitate. You can precipitate out, crystallize, and isolate enzymes in the form of crystals. This is physical property. Molecular weight, huge molecular weight, another physical property. And enzymatic activity is also another physical property. You can see. And this is the diagram that shows physical property. Of course, one property is repeated here. I took this from your textbooks, the new textbook. Precipitation, molecular weight, enzyme activity, biological catalyst, being biological catalyst, and enzyme activity. I don't know that difference here. Denaturation, solubility, and colloids. Don't forget these physical properties as far as your book is concerned, of course. When it comes to physical properties, again, repetition, denaturation, solubility, colloidal nature, precipitation, high molecular weight, and enzymatic activities are biological catalyst. The fact that they are being biological catalysts is also included under physical properties here. And denaturation is the process of breaking the intra and intermolecular non-covalent bonds that distort the shape and active site of the enzymes or proteins. You see, denaturation. Intramolecular, that means inside the molecule. Intermolecular, just between molecules. non-covalent bonds that distort the shape and active site. This is when non-covalent bonds that make up the three-dimensional shape of that globular protein are destroyed, usually irreversibly destroyed, so that the protein or the enzyme loses its three-dimensional shape, and also it loses its function. Also, if it loses its three-dimensional shape, of course, it loses also its catalytic activity, and therefore denaturation simply the loss of the three-dimensional shape of a protein or an enzyme, specifically an enzyme here, so that it also loses its biological role or biological function. Enzymes are denatured by, these are denaturing agents, agents of denaturation. When it comes to human enzymes, above 40 degrees Celsius, high heat, of course, there are enzymes like enzymes of the extreme temperature microbes, the archaea adapted to live in extreme temperatures. There are do have enzymes that can work efficiently, comfortably at more than even 100 degrees Celsius. When it comes to human enzymes, of course, high heat, above 40 degrees Celsius for human, is one agent of denaturation. And alternation in the pH change, too low or too high, beyond the optimum temperature, pH. Heavy metals, and high salt concentrations, high solvents and other reagents can cause denaturation. You see, high salt, these are chemical agents. Heavy metals, high salt concentration, solvents, other reagents can denature. These are known as agents of denaturation. Now, more on protein denaturation in detail, if you like. Of course, protein denaturation in general, not only proteins, but not only enzymes, but proteins in general. Proteins undergo denaturation. Proteins are held in their native conformation by a combination of forces. You can see what are these forces that hold the polypeptide chain to be folded in a three-dimensional, unique, specific shape. One is hydrogen bond, ionic interactions, disulfide bridges, if there are sulfur-containing amino acids especially, and hydrophobic interaction, if there are hydrophobic amino acids which are not water-loving. They come together in the three-dimensional shape of the enzyme of the protein, so that they contribute to the three-dimensional shape. Therefore, don't forget these bonds or interactions, which are very important to form the three-dimensional shape of an enzyme or protein in general. And changing the 3D shape of or configuration of protein is by disrupting these sources. It's called denaturation. If you disturb by external forces like temperature, pH, and some chemical agents, these interactions or bonds, so that you can change the 3D shape of the enzyme or the protein, it is known as denaturation. Thus, denaturation is a phenomenon that involves transformation of a well-defined folded structure of protein formed under physiological conditions, normal conditions, to an unfolded state under non-physiological conditions by changed temperature, changed pH, even some chemical agents. Denaturation results in a loss of protein activity. If it loses its shape, it loses function. Remember, since the native conformation is usually the most water soluble, the native, the natural, the normal structure conformation is water soluble. Disrupting this natural conformation causes changes in solubility and frequently results in the formation of a solid in the solution. It solidifies, it loses. What will happen if you boil egg? Imagine the liquid, the unboiled unboiled raw egg, the protein that surrounds the yolk is fluid. The egg white is fluid. After boiling, exposure to high temperature, it becomes solidified. Can you reverse it? Of course not. Reversible change in the three-dimensional shape of the protein, the albumin protein, made it to solidify. Therefore, usually denaturation solidifies the protein because it loses its natural three-dimensional shape. You can see here the protein at the top is with its natural conformation, natural shape, spherical, globular, When it denatures, it has lost its three-dimensional shape, the spherical globular nature. Agents of denaturation. If we talk about denaturation, what causes, of course, it's redundancy. What causes denaturation in detail here? A change in the structure of protein can be caused by a variety of factors, of course, known as denaturing agents. These include physical agents and chemical agents. And from physical agents, look, heat or high temperature, it could be pressure. High pressure can cause denaturation. Irradiation, the like UV radiation and some ionizing radiations, high energy radiations, and also sound waves. Surface forces are also responsible as physical agents to bring about denaturation. But the most important denaturing agents that we should know are high temperature, from the physical agents, high temperature here. And from chemical agents, of course, chemical agents include acid, low pH, alkaline, high pH, organic solvents, and heavy metals. As I have said, you have to consider change in pH also as one agent. These are chemical agents known as. Um, Enzyme solubility, the other property, we have said a little extra on denaturation. Now, the second property, physical property, enzyme solubility. Solubility is a property of enzymes that allow them to be dissolved in water, salt, dilute glycerol, and alcohol, causing denaturation. Of course, if you dissolve them in solvents which can cause denaturation, the protein will be denatured, it will be solubilized. If it is in water, it's natural, normal solvent, water, and optimum concentration of salt, not extra concentration of salt. It's soluble without losing its three-dimensional shape, functional. Enzymes are soluble in water, sodium chloride, dilute glycerol, and alcohol. Yes. The other property, catalytic property. The biocatalyst property is the activity of enzymes in which very small quantities or a small amount of enzyme is enough to convert a large quantity of substrate and remain unchanged after the reaction. You see, catalytic property means they are used to speed up rates of reaction. That means they serve as catalysts. When they serve as catalysts, especially these highly efficient catalysts enzymes, they are required very small amount. Plus, the other property of catalysts is they remain unchanged at the end of the reaction so that they can be used now and then until the proteins are losing their three-dimensional shape due to the wear and tear after long time of service, of course. The other property, enzyme activity. Enzyme activity, of course, mechanism of enzyme action strongly depends upon the factors like enzyme activity. When we say we have to consider what factors affect the enzyme activity: temperature, pH, substrate concentration, enzyme concentration. All these affect the activity of enzyme. Therefore, when we say enzyme activity as physical factor, we are mentioning what factors, environmental factors affect enzyme activity. And these are the four important: temperature, pH change, substrate concentration, and enzyme concentration, of course. How? Of course, enzymes work at specific uh, uh, temperature range, which is known as optimum temperature range, where they are most efficient. Again, pH range, they work at specific pH range, where they work at most efficiently. Substrate concentration, as we increase the substrate, the rate increases until the fixed number of enzymes reach saturation point, of course. After saturation point, the rate becomes constant. The graph becomes horizontal, of course. You remember the sigmoid, I mean the hyperbolic graph, of enzyme substrate, enzyme rate versus substrate graph. The other is enzyme concentration. As we increase the enzyme concentration, the rate increases indefinitely as far as excess substrates are available. Enzymes show the highest activity at optimum temperature. For humans, 37 degrees Celsius is the average pH for most enzymes inside cells. for most enzymes in our fluid, like blood, it is around neutrality. That is a physiological pH. Except in some areas, like the stomach, our body is almost neutral, around 7. Go to the cytoplasm of cells, go to cerebrospinal fluid, go to circulatory blood, the blood. I remember blood pH is 7.35, I mean 7.35 to 7.45. Almost this is the physiological pH, around 7, the neutrality. A low concentration of enzyme and substrate will slow down the enzyme reaction. Enzyme reaction, yes, low concentration of enzyme substrate here. This is, I think, a mistake. A low concentration of enzyme. If the amount of enzyme is limited, of course, yes, the rate slows down as far as we have excess substrate. If low concentration of enzyme, if the enzyme concentration is low, as we are decreasing the amount of enzyme, the rate is decreasing, of course. In case of substrate, as we decrease, of course, as we decrease the amount of substrate, the rate decreases, of course. As we increase the amount of substrate, the rate increases, but not always until it reaches saturation point. I've told you this, until it reaches saturation point, where the rate becomes constant and the enzyme, the reaction has reached Vmax for a fixed amount of enzyme, of course. Otherwise, low concentration of enzyme means, yes, rate is low. Low concentration, of course, yeah, low concentration of substrate means low rate. On the other hand, a higher concentration of enzyme and high substrate will result in faster enzyme activity. That's clear. Of course, here also, higher substrates means as far as we are increasing the enzyme amount and keeping the substrate increased, will increase the rate indefinitely. But given a fixed amount of enzyme, but excess substrate, the rate first increases and the graph reaches plateau. That's constancy because of the same event that is known as saturation point, where all enzymes are engaged with their full efficiency. Increase in substrate after saturation point will not bring increase in the rate because that is the ability of the fixed number of enzymes. Colloidal nature. This colloidal nature of enzyme is a tendency of having little or no cross the same permeable membrane due to the large size or high molecular. This is simply it's very huge molecular weight so that it can pass the semi-permeable membrane. Enzymes behave as colloids due to their large size or high molecular weight, of course. As a result, the enzymes have a little or no tendency to dialyze or cross semi-permeable membrane. Remember, we do have many different types of proteins in our plasma proteins like albumin, and many immunoglobulins, many. When blood goes to the liver, I mean the kidneys, to be filtered through renal arteries, uh, in the filtering units, the glomerulus. That means nephron glomerular region. Uh, as far as you have healthy kidney, only the water, the salts and metabolic wastes, and the amino acids and glucose. These are only filtered and enter into the Bowman's capsule. The plasma proteins will never be forced to come out of the glomerular filtration unit at glomerulus because they are bulky, they are very huge, they are colloidal. Enzyme precipitation, that is, enzymes precipitation is the separation of enzyme for analysis using different aqueous or ethanol solvents. You can precipitate for diagnostic purpose in healthcare facilities or in clinics or hospitals. You can undergo enzyme precipitation so that you can study their morphology, their chemistry, chemical analysis, for diagnosis of specific diseases related to the enzyme. And therefore, enzyme precipitation is one area of application of medicine for diagnosis, and you can precipitate out. Acidic or alkaline solution can cause enzyme precipitation, as the enzymes are amphoteric. That means they have both acidic nature, alkaline nature, possess amino and carboxylic acid group towards the end of the chain. This is the amphoteric nature. They have positively charged region, which is the amino side, a negatively charged region, which is known as carboxylic side. The negatively charged region is the basic region. The carboxylic acid side chain is the acidic region, so that enzymes do have basic region and acidic region. That's why they are known as, they act as acids as well as bases, which gives them the amphoteric property. Ethanol and a high concentration of inorganic salts like ammonium sulfate facilitate enzyme precipitation. Of course, I'm sorry, enzyme precipitation. Molecular weight. Molecular weight is enzymes are large protein molecules possessing a polypeptide chain of various amino acid sequences. This is also another physical property, therefore huge, many thousands, like 10, 20,000, 30, 40, 50,000 atomic mass unit, of course. Nearly 200 to 300 peptide bonds hold the amino acid together. Therefore, enzymes have a high molecular weight, much to nearly 200 to 300 peptide bonds. Peptide bonds. One peptide bond holds two amino acids together. Two peptide bonds hold how many? Three amino acids together. Four peptide bonds hold four amino acids together. Therefore, 200 peptide bond means how many amino acids are joined together? Subtract 1 from 200, 199 amino acids. If 300 peptide bonds are there, the number of amino acids is 300 minus 1, that is 299 amino acids joined by 300 peptide bonds. There is huge molecular weight. Thank you very much, and thank you very much. Please don't forget subscribing, sharing, and liking my videos. Uh, Thank you very much.